Lockdown is gradually easing, but we still have a long way to go. Beyond the confirmed cases, it's estimated that around 3 million people in the UK could have had the virus. But the big question is, are any of those people now immune?
Science develops by learning from lessons of the past. Now, with this modern virus, we have no idea what the long-term impact of it is on our immune system. To learn that, we have to go and look at studies done more than 30 years ago.
The work of a government research post that closed in 1989 is being dusted off.
Boffins locked in battle against the big sneeze, waging the Cold War on different fronts.
Set up in Salisbury after the war and nicknamed Colditz, the Common Cold Unit aimed to find treatments and vaccines for common respiratory infections. A number of viruses that cause colds were first identified here, including, in the 1960s, coronaviruses. Professor Wendy Barclay was a researcher there in the late 1980s.
This was a sort of hospital/research centre that was set up to investigate infectious disease. You can't really use human guinea pigs, but they decided that common colds were okay. You could infect someone to study what happened.
More than half of colds are caused by a family of viruses known as rhinoviruses. Wendy wanted to see how our immune system responded to these most common of common colds.
I had a group of lovely volunteers who came and allowed us to infect them. And then we asked them to come back, and we gave them exactly the same virus. And the volunteers who'd been previously infected the year before, they were immune from the virus.
But up to a third of common colds are caused by coronaviruses. To test their immune response, researchers infected volunteers with a coronavirus called 229E. When they were exposed to the virus again, the results were very different.
Volunteers, when they came back, got reinfected. They had virus in their nose, which suggests that they might have been able to pass that virus on to somebody else.
And did they get sick?
They didn't get sick, and that was great, but we do have to worry about silent infections if people get reinfected and then can pass their virus on and don't even know they're sick.
Whilst the study only looked at common cold coronavirus, there's evidence that SARS also doesn't bring about a long-lasting immune response. One study showed antibody levels declined over 2 years after infection. And early evidence on today's coronavirus suggests it doesn't necessarily provoke a strong immune response either, though it could vary a lot from person to person.
Here we can see a recent study from China, and you can see people who've made a very healthy antibody response. But down here, you've got some people who haven't made any antibody at all. My suspicion is that this group of viruses, the coronaviruses in general, have in their armament a way of interfering with the way the immune response would normally work, so that the antibodies get made more slowly than usual, perhaps fewer antibodies than usual, and they don't get maintained for as long as we might usually see with a different virus, where we do sometimes see this long-lived immunity.
Antibodies are only one part of a complex immune response, but this could have a big impact on herd immunity. The idea that once enough people in a population are immune, the virus cannot spread.
The danger presumably is looking at that. The herd immunity could fade away.
Yes, so that we can never achieve that high level of where enough people all at once have got protective antibodies that the virus would be stuck and have nowhere to go.
Fortunately, there is another approach to stopping this outbreak. Develop a vaccine.
It's taken nearly 8 years' research to get this vaccine, and it's called a live vaccine. A specially weak form of the virus is grown in cells of rabbit kidney.
Vaccines are estimated to prevent more than 2.5 million deaths every year from infectious diseases like measles. They can induce longer-lasting immunity than we might get from an infection. more than 100 groups around the world are racing to develop a coronavirus vaccine. I want to find out how a process that normally takes years is being fast-tracked safely.
I'm on my way to Portendown in Wiltshire. It is one of the most secure testing facilities in the UK.
Researchers here work on some of the world's most dangerous diseases, like anthrax, Ebola and the plague.
But all that has stopped, and we've diverted all our attention and capability for coronavirus.
Professor Miles Carroll has run trials to make sure 2 candidate coronavirus vaccines are safe for use in humans. And that meant trialling the vaccines on animals first.
We have started vaccinating macaques and ferrets with candidate vaccines. We wait a certain amount of time after vaccination, then we challenge with a known amount of virus.
Animals are challenged by exposing them to the virus, then they're evaluated.
We look for differences between the vaccinated animals and the unvaccinated animals to show A, the vaccine has worked, but importantly B, to address any safety concerns.
The need to ensure the public can be confident vaccines are safe is highlighted by events in America in 1976.
No one knows exactly how serious this threat could be.
The country faced an outbreak of a deadly new swine flu virus.
I am asking each and every American to make certain he or she receives an inoculation this fall.
40 million Americans were vaccinated. A small number of people developed a rare neurological condition called Guillain-Barré syndrome, which can lead to paralysis. Research showed those who were vaccinated had a small increased risk of developing GBS. The exact reason for this link remains unknown. Nevertheless, public confidence in vaccines was shaken, showing why robust safety trials are vital, even in the middle of an outbreak. At Portendown, one focus of the safety trials is making sure a vaccine does not lead to rare immune system complications that can cause serious disease after exposure to the virus.
With a related vaccine, SARS-1, in some instances in animal models, there was an indication that you did see enhanced disease, but they were different types of vaccines that we're testing here today.
Animals in the trial will have their lungs checked for possible damage.
With the COVID-19 vaccine, are you seeing any sort of bad things happening?
So we've only vaccinated the animals and they look completely and utterly healthy, and we won't be able to measure any effect until after we challenge the animals.
So far, vaccines that you've been testing out on animals appear to be safe?
They certainly do.
Because vaccines are given to healthy people to protect them from infection, establishing they are safe is paramount.
Risk-benefit ratio, someone dying of cancer, you would risk giving them a more toxic drug to save their lives. But if you're vaccinating a healthy individual, then you have to have something that is extremely safe and will do no harm.
One of the candidate coronavirus vaccines Portendown has been testing is developed by Oxford University.
The first human trial in Europe of a coronavirus vaccine has begun in Oxford.
The vaccine has been allowed to start human trials after successful safety tests in America.
The normal way we do this, it takes us several years to get to the point of having a vaccine that would actually be even considered for widespread use.
And now the group is gearing up to produce around 1 million doses of the vaccine before they know it works.
We've started moving to doing real clinical manufacturing at industrial scale very early. We want vaccine to be ready as soon as the clinical trial shows that the vaccine can be safe and work in people. But to have the vaccine ready then, we need to start months and months in advance. And we're hoping, if everything goes really well, we might have some vaccine ready to use in perhaps the early autumn.
At that point, regulators might grant an emergency use of the vaccine that would allow the human trial to scale up to potentially vaccinate tens of thousands of people.
It would be initially relatively restricted. Healthcare workers, maybe people living in care homes, perhaps older people with underlying lung diseases. The people who've got the most to benefit from the vaccine would likely be the first to receive it.
The Oxford vaccine is just one of over 100 being developed by the world's leading vaccine scientists. Now, in the past, they focused on diseases like HIV, influenza and Ebola, and now they're bringing all of those different techniques that they've developed to the new coronavirus. Vaccines work by stimulating your immune system to produce these proteins called antibodies that bind to the surface of a virus and neutralize it. All of the vaccines you've ever had fall into one of two broad categories, live ones and dead ones. Dead vaccines are the simplest. In this case, a protein from the new coronavirus is made in the lab and then injected into the body. It's safe, but in general, vaccines like this don't generate such a long-lasting immune response, and boosters are often needed. The other type that a few different groups are working on is live vaccines. A weakened form of the coronavirus itself is injected. Now, these vaccines create a strong immune response, and vaccines like this have been used for chickenpox, smallpox, measles, mumps and rubella. But we often can't give live virus vaccines to older people or to those people with weak immune systems. In other words, the very people at greatest risk of COVID-19. Now, dozens of other groups are working on technologies which have never been used widely before in human beings. So the group at Oxford are using an approach called the viral vector method. They take a virus that normally infects chimpanzees. They genetically modify it so it can't reproduce or spread. And then they also add in a gene here for the new coronavirus spike protein. Now, when this chimp virus enters cells of the human immune system, the coronavirus spike protein is made, and it goes to the surface of those cells where it's recognized by other cells of the immune system, and that triggers them to make antibodies against the COVID-19 virus. And around 25 groups around the world are using this approach. These technologies are impressive, but there's no guarantee that any of the approaches will work. And even if one does work, and it can be safely mass-produced for the general population, it could still take well over a year. For right now, we are going to have to live with the virus because the lockdown measures have not eradicated it. So as the country is starting to open up, how are we going to balance that greater freedom that we all want against the risks of a second peak?